Our sense of smell is profound. We can smell and remember tens of thousands of different odours.
I like lavender and I like strawberry smells.
My favorite smell is ginger.
I like it here because I like the smell of the flowers, the lilies that are down here. Even the fish smells good.
Oysters. They smell like it's dirty, very dirty. Smell like the bottom of the ocean floor.
The mystery of how we smell has been solved by this year's Nobel laureates in physiology or medicine. They are Linda Buck from the Fred Hutchinson Cancer Research Centre and Richard Axel from the Howard Hughes Medical Institute.
My laboratory has been interested in how the brain represents the external world. After all, the external world consists of a very rich array of aspects of reality, and the richness of the external world has to be represented in the brain by simply a collection of neurons.
Linda Buck was born January 29, 1947, and is currently a professor at the University of Washington. She received her bachelor's degree in psychology in 1975 from the University of Washington and her PhD from the University of Texas Medical Center in immunology. Richard Axel was born July 2, 1946, and is a molecular biologist and professor at Columbia University in the Department of Neuroscience. He received his Bachelor's of Arts in 1967 from Columbia and his medical degree from Johns Hopkins University in 1971. Axel and Buck met in 1990 when Buck was completing her postdoctoral work at Columbia in Axel's lab. Buck was interested in the area of odor detection and the cloning of genes, while Axel was interested in perception and how our genes could explain the way the world is represented in our brains. Using their individual areas of interest, they cloned the olfactory receptors of rats and discovered that they belong to a large family of G protein-coupled receptors. Using rat DNA, they found that this gene family was made of 1,000 different genes that gave rise to olfactory receptor types. These receptors are activated by odorant molecules that form an odorant pattern that allows humans to detect 10,000 smells from memory. They later found that each olfactory receptor neuron only expresses one kind of olfactory receptor, and the input from all the neurons expressing the same receptor is collected by the olfactory bulb. It was this discovery that led Axel and Buck to win the Nobel Prize in Physiology or Medicine in 2004.
The first challenge faced by Axel and Buck was to find the receptors in the nose which are crucial to smell.
It had been proposed that there were protein receptors that recognize odorants, but no one had been able to find them. So that was the initial work that I did in Richard Axel's lab, searching for those receptors.
After 3 years, Axel and Buck found the receptors. They knew that different smells arose from different molecules. However, they needed to find out how the combination of receptors with these molecules actually produced the experience of smell.
And a mystery was how it is that these different structures are perceived as having different odors. These two molecules have almost identical structures, and yet they have different smells. One smells like pear, and the other one like banana.
Smell functions very similarly to our other senses, such as taste or vision, but differs in several key ways. Odorant molecules are released by any substance characterized as volatile, meaning that it can easily be transformed into a gas. This is why non-volatile materials, such as stone or steel, have no discernible odor. These odorants are directed up the nostrils until they make contact with the olfactory epithelium, 1 square inch of mucous membrane located inside the nasal cavity. Odorants bind to chemoreceptors on the epithelium, causing an electrical impulse to travel to the olfactory bulb. Axel and Buck made history when they discovered that there are over 1,000 different specialized receptors in the epithelium that respond with varying intensity to any given odor. By comparison, there are only 3 different photoreceptors in the eye that transpose light into our visual world. In addition to identifying the many specialized chemoreceptors located in the epithelium, Axel and Buck explained how the impulses are translated into discernible odors. Each receptor type sends its impulse to a specific region on the olfactory bulb called a glomerulus, which in turn passes the impulse onto other parts of the brain. The different combinations of activated glomeruli translate into different recognizable smells.
Our sense of smell is an important tool for navigating the external world. This system is so important, in fact, that 1,000 different genes, or 3% of the human genome, are devoted to the olfactory system. The use of smell enables us to perceive danger, such as smelling smoke from a fire or avoiding food that is spoiled. The sensory system also plays a large role in our ability to taste, which is why things might taste bland when you are congested. Smell is also closely related to our memory and emotions. Particular scents can be associated with events that could lead to a feeling of nostalgia. This relationship is used by advertisers to market their products. For example, a realtor that bakes cookies before the open house, or the use of scent in a coffee commercial.
One more.
When it seems like nothing else will, the fresh mountain-grown aroma of Folgers coffee can really get you out of bed.
Kiss the kids are up. The best part of waking up is Folgers in your coffee.
Our sense of smell was once the least understood of the senses, but now through the research of Axel and Buck, this sense has become less of a mystery. Potential applications for the information that their research has discovered about the olfactory system could lead to science to discovering ways to prevent diseases.
The discovery of how we smell has huge implications for combating the spread of disease.
Efforts are being made to approach malaria by a greater understanding of how the mosquito, which carries the malarial parasite, uses its nose, its antenna, to detect the human host. By understanding how it is that the insect recognizes the odors of man and what the nature of those odors are, one can develop extremely powerful inhibitors of the olfactory system, which might be the most effective insect repellents available.
By explaining smell, the laureates have given scientists a potent weapon against disease and brought us closer to understanding the pleasure in odors.